This slide shows the row-column alignment from the input sequences, while this slide shows the mechanisms of multiple sequence alignment. As we show, the conserved regions help to identify functionality. On the other hand, this slide shows the difference between high score and significant after multiple sequence alignment. Whereas, this slide shows the percent sequence identity, which describe invariant of two nucleotide or amino acid sequences. As we refer, there are mismatch sequences, and indel mutation, while the identical sequences are 70%. On the other hand, this slide refer to the score of 8 sequences after alignment. As we show, the first example has 6 identical sequences to all 8 sequences, while the second example has 3 identical sequences to all 8 sequences.